It's a great pleasure to participate in the webinar about the immuno-oncology virtual event. Today, my topic is the strategies and the points in the circular RNA process development. Next, I will introduce from the following four parts: the overview of the circRNA, the design and the manufacturing of circRNA, the quality research of the circRNA, and our company induction. First, let's see what is the circRNA. The circRNA are a class of covalently closed RNA molecules which lack the 5 and 3 terminals. Its formation comes from the backsplicing of the pre-RNA, which allows the downstream splice donor to join backwards to an upstream splice acceptor, while the linear RNA formed by the canonical splicing. Because the circRNA don't have the 5 and the 3 terminal, protect them from the digestion by the exonucleus. So the circRNA is more stable than the linear RNA. And then let's see some breakthroughs and progress in the circRNA development. The circRNA was first discovered in the 1976, and then in the 1979, it is also discovered in the eukaryotic cells. In the following decades, the PIE systems, the IRES, and others like the purification and the modifications are successfully detected and well studied. Researchers optimized the untranslated sequence in the circRNA for the efficient and accurate protein translation. These developments have led to the breakthroughs in the artificial in vitro synthesis of the circRNA and made it possible as a biomedicine strategy. In the recent 2022, the engineered circRNA vaccines are used for the first time in the treatment and the prevention of the COVID-19 and the melanoma. Here, we list some difference between the circRNA and the linear RNA in the preparation, the storage, the biosecurity, delivery, antigen encoding, and the application. In terms of the biosecurity, modified mRNA exhibits low immunogenicity, whereas the circRNA can exhibit low immunogenicity without the modification, and has even lower or no immunogenicity after the modification. And the circRNA has the prolonged antigen coding tolerance due to the high longevity of the circRNA. In terms of the application, in addition to being used in the vaccines, encoding the adjuvant antibody and the protein replacement like mRNA, the circRNA can also be applied to sponges and the endogenous circRNA mimics. Let's introduce the biomedical applications of the circRNA, including act as the RNA partner, the sponges of the microRNA, sponges of some proteins. Also, it can act as the antisense RNA or boosters or the inhibitors of the initiate immune response. Furthermore, the circRNA can also be utilized for the protein expression, particularly in the context of the protein replacement therapy, or it has the prolonged peptide expression. In addition, the circRNA can also act as the pathological targets and the biomarkers that can be applied in different kinds of human cancer. Currently, the in vitro synthesis of the circRNA is primarily achieved through the following three methods: by the chemical or the enzymatic ligation, which relies on the complementary template to bridge the two terminals, and the ribosome ligation includes the group 1 and the group 2 intron, which catalyze the splicing. Our preparation process for the circRNA includes the sequence design, plasmid preparation, in vitro transcription, circRNA purification, LNP encapsulation, and ultimately the formation of the circRNA drugs. Now, let's take a look at what RNA customization service we can provide. Our offerings covering 4 main parts: the RNA design, RNA drug substance, RNA LNP encapsulation, and the quality research throughout the entire process. In the RNA design, we specialized in the circlization optimization, including the circlization element design, translation element design, and the signal peptide design. And in the target gene optimization, we specialized in the structural design and the codon optimization. And in the second part, the RNA drug substance, we can provide the preparation of the DNA transcription templates, which include the construction of the plasmid templates and engineered bacteria, establishment of engineering bacterial seed banks, preparation of plasmid amplification and linearized templates. And in the preparation of the RNA, our expertise in the in vitro transcription, modification of the nucleotides, the cellization of the circRNA, and the purification of the circRNA. We move on to the RNA-LNP encapsulation. Our capabilities encompass the LNP formulation, screening, and the preparation of lipid nanoparticles to ensure the efficient delivery of the RNA therapeutics. Regarding the quality research in the entire process, there will be a separate chapter to introduce it later. In the sequence design, we will showcase our outstanding abilities, circularization strategies for the circRNA, translation element design, and the secondary structure design. First, we have developed a novel circularization strategies, which we refer to as the isopi. In this slide, we present the details, design schematics for the various circularization elements that different from the clean pi and the anapi, and we tested the GFP expression at the cellular level and found that the isopi 10 and the isopi 11 exhibit the enhanced and longer expression compared to the other circularization element. Furthermore, we also screened a set of internal ribosome entry sets, which means the IRES, and identified some highly expressed IRES. The IRES are the specialized RNA sequence that can directly recruit the ribosomes to the internal position of the RNA and initiate the translation. The well-known IRES elements that are the commonly utilized for the robust expression include the CVB3 and the EV29. In our screen results, we got a YX6 IRES, which exhibits the enhanced GFP expression compared to both CVB3 and the EV29. And also, we got the YX1 and the YX2 expressed similar like the CVB3 and the EV29. Additionally, we have also attempted to optimize the secondary structure of the target gene. During the optimization process, we mainly focus on 2 indicators, the CAI and the MFE. CAI, which stands for the codon adaptation index, indicates that the higher values correspond to a higher preference for the specific codons, while the MFE represents the minimum free energy. with lower values suggest a more stable structure. We established different combination methods using these two indicators. For the CAI, we designed 3 levels: the high CAI, mid CAI, and the low CAI. And the MFE, we have the 4 levels: the low MFE, mid MFE, lowest MFE, and the high MFE. And we combine different levels together. We can see that the CDS designed with the mid CAI and the lowest MFE has the compact secondary structure. It not has so much loops as the CDS designed by the low CAI and the high MFE. So it is more stable and not easily digested by the endonucleus. We compared the expression of the luciferase with different designs and find that with the mid CAI and the lowest MFE, it has the highest gene expression. The second part is the RNA drug substance, which include the preparation of DNA transcription templates and the RNA. Our plasmid and template preparation process is well established. The plasmid yield is about the 0.6 to 1 gram per liter, and the supercoid ratio is guaranteed to be higher than the 85%. We can see in this case, before the purification, the crude plasmid purity is about 60%, and after the purification, the plasmid purity reached about the 98%. Besides, the high-quality templates were also important, not only for the IVT yield, but also the purity. Our linearized plasmid templates is higher than the 95% purity. For the in vitro transcription, we have T7 enzyme library, which contains the wild-type T7 and a variety of the T7 mutant, and we have optimized the process for each T7 enzyme. In addition, we found that the different sequence exhibits the different T7 enzyme preference. For example, the 3 sequence in the left figure, the mutant 3 T7 enzyme has a better cyclization rate than the wild-type T7 enzyme, while for the sequence in the right figure, the wild-type T7 enzyme is more effective than the mutant 3 T7 enzyme. So, for each individual sequence, we offer the screening of the T7 enzyme and their corresponding process in our library and identify the optimal IVT process. In the circRNA purifications, we have 2 strategies, the RSR purification and the chromatographic purification. The RSR purification are suitable for the short circRNA and the circRNA with the loose secondary structure. In parallel, we have also developed a chromatographic purification method, which the purity of the circRNA can be increased to more than 90%. And this technique is without the restriction of the sequence length and suitable for the circRNA with very compact secondary structures. RSR is a exoribonucleus that can digest the linear RNA from the 3-terminal to the 5-terminal, but not digest the circRNA. In the circRNA produced by the IVT, the main contaminants are the precursor RNAs, the nicked RNAs, and some introns. By employing the RSR to enzymatic digest these linear impurities, we can isolate the circRNA with the high purity. This is a case shows the purification effect of the circRNA by RSR treatment. We use 2 HPLC method to check the purity of the circRNA before and after the RSR treatment. And both methods show that After the RSR treatment, the circRNA purity increased from about 60% to over the 90%, and the impurities peaks include the NixRNA, introns, and the NTP almost disappear after the RSR treatment, indicating that the RSR plays an excellent role in circRNA purification. Next, let's see a new method we developed to purify the circRNA by the chromatography. The purification of the circRNA through the chromatography has been a great challenge, especially for the separation of the circRNA and the ncRNA, which have almost the same molecular weight and the charge properties. So it is difficult to achieve effective separation through the conventional chromatography method. And now we have successfully developed a novel chromatography approach that can successfully separate the circRNA with the impurities. From this ACTA spectrum, we can clearly see two distinct peaks with a high degree of the separation. By analyzing the samples collected from the respective peaks, we can conclude that the front peak is the circRNA and the back peak is the impurity RNA. This is the purity result of the sample collected after the chromatography. We also use two HPLC method to check the purity of the circRNA. We can see in the HPLC-2 method, the front peak is the circRNA and the back peak is the impurities. We can see that after the chromatography purification, the purity of the circRNA is significantly increased, and the impurities has been largely separated out. And also the electrophoretic evidence further confirm the substantial enhancement of the circRNA purity, and the majority of the impurities have been effectively eliminated. Additionally, we assessed the GFP expression at the cellular level 4 days after the transfection to compare the circles purified by the chromatography and the circles purified by the RSR treatment, and find that the expression level of the circRNA purified the chromatography expressed almost twice as the circRNA purified by the RSR. So, indicating that the higher purity of the circRNA may yield more effective expression. The third part is about the RNA-LNP encapsulation, which include the LNP screening and the preparation of the nanoparticles. This is the RNA encapsulation process. The lipid mixture and the nucleic acid solution are combined through the microfluidic mixing to form the RNA-loaded lipid nanoparticles. And following the buffer exchange, parameters such as the encapsulation efficiency, the particle size, and the zeta potential can be assessed. Here, we present some data from the encapsulation process. In addition to the commercial cationic lipids, the SM102 and the ALC0315, we have also developed the liver-targeted cationic lipids. And the parameters in all these cases shows a good quality. The post-encapsulation particle size can be finely controlled to an average about 100 nanometers, and even can be reduced to 80 nanometers, and the PDI remains below 0.1, indicating a highly uniform particle size distribution. The encapsulation efficiency maintained above the 90%, and some can reach the almost 100%. And the maximum concentration of the encapsulation substance reaching up to nearly 2 milligrams per millimeter. Furthermore, the encapsulation efficiency, particle size, and the PDI of the formulation remains stable after storage for 4 weeks at the room temperature. And we also inject the mouse by the circRNA like LNP with different types and concentrations of the cationic lipids. And we discovered that there was a higher level of the expression when 30 micrograms of the RNA-LNP encapsulated with the SM102 were injected, and this expression could be maintained for about 8 days. This part contains 2 case studies. The case 1 is about the comparison of the circRNA and the linear RNA, and the other case is the function of the iPSC transcription factor circRNA. In the first case, we compare the expression level of the linear RNA and the circRNAs, and we find that the linear RNA express higher than the circRNA at 24 hours, while as the time goes on, the protein expression of the circRNA is significantly higher than that of the linear RNA, particularly after the 3 days. And both the cell fluorescence and the Western blot shows the same result. And besides, we also screened the stock buffers of the circRNA, and we found one buffer can well stock circRNA at the room temperature for about 1 month. In the second case, the circRNA of the iPSC transcription factor OSK-mLN were transducted to the HDF, and successfully result in the iPSC-like clones capable of the stable self-renewal. This part is the quality research, includes the testing and release for the plasmid DNA, circRNA drug substance, and the RNA-LNP drug product. This is the testing and release for the plasmid DNA, which contains the attributes and the method of the identity, content, purity, impurity, and the safety of the plasmid. Similarly, here are some testing and release that can provide for the circRNA drug substance. And here are some testing and release that we can provide for the RNA-LNP drug product, which also contains the identity, content, integrity, purity, safety, potency, and some others. In summary, we can provide the full process of the CMC service from the small-scale research, which includes the RNA sequence design, the small-scale process research, and the small-scale simple preparation from the 1 to 100 microliter. And we also provide the pilot scale research, which contains the RNA drug substance process research from 0.1 to 10 liter, and the RNA-LNP encapsulation research from the 1 liter to 50 liter. Besides, we also provide the stability research and the quality research. In addition, we also can provide the clinical trial application and the simple preparation, which includes the process transfer, preparation of clinical trial sample, and the compilation materials. At last, I want to introduce my company, the YXgene. We have more than 20 years of the CDMO service experience, and we have more than 20 IND submission projects, and 13 clinical approval, 2 NMPA or FDA clinical approval, and more than 20 times files audit. We provide CDMO service for gene therapy and new vaccines development. We focus on the virus vectors, oncolytic virus, and the mRNA product. And we offering the histologic solutions in the process development and the analytical method, the IND enabling CMC, clinical, and the commercial contract manufacturing. Cooperate with us and perfecting your future. And besides, we are eagerly recruiting for a global BD consultant and looking forward to your journey. You can contact us through the following email address. Okay, thank you.